Good day. My name is Franco Lori, and I'm the director of the Research Institute for Genetic and Human Therapy, RIGHT, which is a US-based non-for-profit research organization with operation in Europe. And today I'm here to convey the following message. A cure for COVID-19 is possible, but it has to be based on at least 3 rules. Number 1, it has to be based on a cocktail of drugs as opposed to single drugs. Number 2, it needs the proper team, and I would like to convince you that we are the team for this job. And number 3, it has to be fast. We cannot measure success by years or months. We have to measure success by weeks. And in the following part of the video, I would like to convince you that these rules are respected within our team. Why are we the right team for the job? When I was a postdoc at NIH in Bob Gallo's lab working on HIV, I came to the following idea. Combine a drug that was specifically designed against HIV with another drug that was not specifically an antiviral, but was acting upon the cell, hosting the virus, and taking out from the cells those essential elements that were vital for HIV to replicate. And it worked. We published back then in Science magazine. Then I left Bob's lab in order to start RIGHT. And we were really focusing on clinics. I'm an MD by education, so my obsession is really how to find a cure. We added a third drug that was one of the very first, actually the very first protease inhibitor. And with these 3 drugs cocktail combination, we promoted and induced the very first functional cure of an HIV-infected patient, the famous Berlin patient, which was then published in the New England Journal of Medicine. So we did it with HIV, we can do with COVID-19. And let me tell you how. How is our approach different from other approaches? And why do we think that we can make it where others might fail? That's a very important issue, and it rotates around the concept of combining drugs. Back in the initial times of the HIV pioneering era of antiviral therapy, we had one drug, was AZT. It was working somehow, but very little. When we combined 2 drugs, we saw better results, but still not enough. And it was only after combining 3 drugs that we started really to see a difference. It was like black and white. Same happened with hepatitis C. The drugs are done singularly, but then delivered in cocktails. And that's exactly what we want to do here. Are there other people trying drugs? Yeah. Mostly singularly or in dual combination. Are other people combining 3 drugs? Perhaps some. But again, the approach is really randomly driven. Do we have a systematic, rigorous, scientifically based approach in order to combine as many 3 drugs as possible? That I don't see. That's what we can do in our lab. And you need to go back to the laboratory to do something like that, because there are so many drug combinations that we can think of it would take simply a long time in the clinics in order to try and figure out whether they work or not. We can do it in the lab, and we can do it in the lab in a large number. And one of the advantages that we as virologists have compared to other branches of medicine is that we can be relatively predictive of the potential success. But we have to combine drugs that are already available, commercially available, and the drugs that are repurposed from other indications towards the cure of COVID-19. Only by combining drugs that are already indicated for other purposes, we can be fast. Because if we had to develop new drugs, it would take years. We don't have that time. But if we combine drugs that are already available, we show that they work in our laboratory system in combination, then we will be relatively fast to then move to the clinics, go through rigorous and well-designed clinical trials, and prove in the clinics also that they work, and prove that the toxicity is acceptable, then can be used by everyone. The timing we are suggesting to achieve the first laboratory results is 8 to 12 weeks. It seems a lot, but the people are predicting that to design a good vaccine is going to take 8 to 12 months, and we all know that a regular drug might take 8 to 12 years. So even if it looks a lot right now, it's really a record time for identifying something that could work against COVID-19. How the drugs might be working? Well, there are several approaches, again, and some of them work by destroying the capsid that the virus needs in order to reinfect the next cell. Some others are really designed to interfere with the construction of the genetic material of the virus, those building blocks that the virus needs in order to replicate. And the third one, as I said before, and this is very important, has to be an indirect inhibitor of the virus. A drug that interferes with the one or more of the mechanisms that the virus relies on when infects a cell. A virus, by definition, is not able to survive without hijacking some of the material that the cell has. And that's exactly where we have to intervene. So it is a three-pronged approach, at least three-pronged approach. And we are talking about antivirals. There are other drugs that are not antivirals that are being used against COVID-19. Those are anti-inflammatory drugs or immunosuppressive drugs that people use in order to mitigate the consequences of viral infection, which results in hyperproliferation, hyperactivation of the immune system. That's a different approach, and the two of them are not alternative. But here is the trick. We don't want to use the drugs in the wrong moment of the infection. Antiviral drugs should be used as early as possible. in order to limit the spread of the virus and the consequences of this spreading. The anti-inflammatory and particularly the immunosuppressive drugs might not be really useful at the beginning of the infection. In fact, by limiting the immune response, the virus might even have better chances to spread. But once the virus has spread, and once the consequences of it result in hyperactivation and hyperinflammation, that's where we have to use those drugs. At that point, perhaps antivirals will not be as effective as they would be if they were given at the very beginning of the infection. They could still have some effect, but again, we have to provide the drugs at the proper time with the proper approach. We need to be cost effective. We are proposing a first part of our research in order to obtain a first indication that some of the cocktails that we are experimenting are indeed valid and should be tested in the clinics for $250,000. If we think about the potential damage of this virus if it goes unattended, which is now roughly estimated in trillions of dollars, we can imagine how cost-effective our research would be. We can do it with a relatively small amount of resources. We can be fast, again, 8 to 12 weeks. We can be effective because we have the right laboratory, the proper facilities that are needed to handle highly pathogenic viruses, the personnel that has been trained with HIV and other viruses, which is excellent and can do the job. We have the experience. We have the proper attitude and thinking towards drug combinations, what might effectively work. And we are very, very motivated to achieve something that in a short time, with little resources, might really change the world of COVID-19. Imagine the consequences. As much as we invest in the lab could be saved and spared to empty hospitals, to fill up squares, to have people gathering again. I think it's worth the try. And thank you in advance for helping us achieving this goal. Have a good day.